That's why the cytoplasm of these cells contain numerous mitochondria. Now we come to the connective tissue proper, to the connective tissue fibers. Connective tissue fibers are elongated structures formed mainly of proteins or formed of proteins, and there are three types of connective tissue fibers: collagen, reticular, and elastic fibers. Collagen fibers are formed within by the fibroblast, for example. Part of synthesis of collagen fibers occurs within the lumen of rough endoplasmic reticulum. Collagen fibers is formed of three molecules of alpha chains. These three molecules of alpha chains, which will assemble forming triple helix. This triple helix molecule is known as procollagen, with both ends containing loose or with both loose ends. Then from the lumen of the rough endoplasmic reticulum, the procollagen molecules will be passed to the Golgi apparatus, where they are packaged into secretory vesicle, and they are released to the extracellular space. Within extracellular space, procollagen peptidase will be acted upon this molecule, removing the loose ends and forming collagen molecules. These collagen molecules will assemble to form collagen fibrils. Then group of fibrils will assemble to form collagen fibers. Collagen fibers could be stained by either Eucein stain, and they will appear as pink wavy bundles, and also they could be stained by special stain known as Mallory stain, Mallory blue. Mallory stain gives them blue color. By electron microscope, collagen fibers shows characteristic axial periodicity. The meaning of axial periodicity, it means that each fibril of collagen or each collagen fibril shows alternating dark and light bands. This alternating dark and light bands occurs due to the special arrangement of the collagen molecules within the collagen fibrils. Within the collagen fibrils, the collagen molecules are arranged in the form of stepwise overlapping pattern. This stepwise overlapping pattern produces an area of overlapping between the molecules of collagen and a gap region. The heavy metals used in the electron microscopic studies will be deposited in this gap region. While the overlap region, there is no gap, so no deposition of the heavy metal. That's why by electron microscope we can found darkly stained area, which is representing the gap region due to the position of the heavy metal, and lightly stained area representing the overlapping region. And this pattern of periodicity occurs at a regular interval every 64 nanometers. To date, there are 28 types of collagen fibers. They have different amino acid sequence, producing different types of collagen fibers, and they are giving orders or numbers according to the time of their discovery. The type 1 is present in the tendons and bone, type 2 present in cartilage matrix, type 3, for example, forming reticular fibers, type 4 in the basal lamina, as we studied in the first term, and type 5 is present in the blood vessels. Different group of diseases affecting collagen synthesis formation is known as collagenopathies. So collagenopathies, it is abnormality in the formation of collagen fibers. Example of these collagenopathies is osteogenesis imperfecta. In osteogenesis imperfecta, the person suffers from a brittle bone, loose sclera, and loosening of the loosening of teeth. And for more details, you can look on Also, sun exposure may lead to accelerated photoaging of the skin or photoaging skin changes due to affection of the assembly of tropocollagen molecules or collagen molecules by the sunlight, resulting in unstable collagen fiber. The second type of collagen fibers is reticular fibers. Reticular fibers are type 3 collagen fibers, but they are surrounded by glycoproteins, and they can be stained by silver stain, as we see here. Silver stain making the reticular fibers appear in the form of thin branched fibers forming a reticulum supporting the internal organs. The last type of connective tissue fibers is elastic fibers, which appear as thin straight fibers and they are branching, and they could be stained by special stain known as by electron microscope. As we can see, elastic fibers do not show axial periodicity, because the axial periodicity is only a characteristic of collagen fibers and reticular fibers, but elastic fibers do not show axial periodicity. They are formed of core of elastin protein, and this elastin protein is supported from outside by microfibrillar protein formed of fibrillin protein. Defective formation of elastic fibers resulting in a syndrome known as Marfan syndromes. Marfan syndrome has many symptoms, and you can look for it in your uploaded textbook. The most important feature or symptom of Marfan or complication of Marfan syndrome is the presence of aortic aneurysm, which is dilated weak area of the aorta that may rupture at any time, and it is a life-threatening condition. Lastly, we come to the examples of the different types of connective tissue. The umbilical cord is an example of mucoid connective tissue. This is a section of umbilical cord. As we see, it is formed of two arteries and one vein. The tissue filling the spaces in between this structure is the mucoid connective tissue. It is formed of mesenchymal cells, undifferentiated mesenchymal cells with long cytoplasmic processes and densely with long cytoplasmic processes, and separated by jelly-like matrix or jelly-like material known as Wharton jelly. Then we come to other type of connective tissue, which is the dense collagenous connective tissue. The example of dense collagenous connective tissue is the tendon. Dense collagenous connective tissue is formed of collagen bundles which are parallelly arranged to each other and separated. These bundles are separated by the nuclei of fibroblasts and fibrocytes, which are responsible for formation of this type of collagen fibers.